Okay, let me introduce what we can do with food metabolism repository. I'll show you three examples. First one is the case of caffeine. As you know, caffeine is contained in tea and coffee. When I searched the exact mass value of the caffeine, I got this result. The strong peaks detected around 38 to 39 minutes must be caffeine because they are detected in tea and coffee. These peaks with the different recension times should be isomers or just noise peaks. But here, the caffeine peaks is weakly detected in honey too. It was very surprised. So I sent an email to the honey company and asked about this possibility, but they said no, it must not be. It is a natural response when I think about a risk of the bad rumor. But with a careful checking of the literature, I found several reports supporting this situation. Some citrus plants accumulate caffeine in their flower and nectar, and caffeine can be detected in honey too. And when honeybees intake caffeine, they strongly memorize the location of the flower. So it is said that the accumulation of caffeine in flower is a survival strategy of the citrus plant by raising the chance of the pollination. This use case showed that searchable metabolism data repository like food metabolism repository is useful for making a good working hypothesis without any specialized background knowledge. The second case is discovery of okaramine. This is a case of research field different from foods. Rhizosphere is a soil region very near to plant roots. In the rhizosphere, symbiosis with soil bacteria is very active, corresponding to the intestinal flora of humans, and the conditions of the rhizosphere strongly affect the plant growth. But the chemicals in the rhizosphere are largely unknown. So we analyzed the soil metabolism in a research project with Kyoto University. We compared 5 soil samples and found some candidates specifically accumulated in the rhizosphere or hairy vetch. It is a green manure-rich plant. In the candidate list, I found a strange name, okaramine, because okara is a waste of soybean produced in the tofu production. So I checked about the okaramines deeply. Okaramines have pesticidal activity and originally found in the culture of a soil bacteria when it cultured with okara as medium. But there was no report for the existence in the nature. So I checked the mass value of okaramine peaks using food metabolism repository, which contains more than 970,000 peaks. I got the result, no hit in the food. This is quite reasonable if the compounds are generated by soil bacteria. This result was a very big drive force for plants into the identification, and we successfully identified the okaramines for the first time from the nature. At present, we are studying about the application of these okaramines as agricultural material. The okaramine case showed that the food metabolism data are useful for any field, and probably vice versa. The last case is the discovery of novel flavonoids using API. APIs, application programming interfaces, are the functions for data acquisition, searching, and so on from the other computational programs. For example, when accessing this URL containing the search parameters, you can get the search result in a text data in JSON format, which is an easy-to-use computer-readable format. API provides the way for higher level of data mining, such as automation of large-scale searching and complex data processing with support of computational programs. The details of APIs are described at the help page of the website. This is an example of use of API to get the sample list. When Python program, you can get this result by writing only 4 lines, very simple. When you get the food IDs from the list, you can use the information in other search. For example, getting a pick list of the food by the food ID. Again, it requires only 4 lines to get the result. Using the API, I tried to discover the candidates of novel flavonoids. Among the peaks with MS3 spectral information, I filtered the peaks with no compound search results and high flavonoid search results. So they must be the candidate of flavonoid-like compounds but never registered in the compound databases. This kind of filtering can be done in a short time using APIs. So I found small number of peaks, only 28 peaks filtered in this condition. So I checked them one by one manually using Food Metabolomic repository website. I will skip here to describe the details of the checking procedure, but I found many interesting candidates in a brief time, such as a candidate of methionine conjugated flavonoids in grapes, and citrus-specific flavonoids with substituent of this formula, C5H9O2N. It corresponds to the proline. This example shows that searchable repository with APIs is powerful for discovering novel compounds. So now you can perform data mining using Metabolome data. It can be used for integrated analysis with other OMICS data for identification of the genes and for co-accumulation network analysis to find the key components of specific biosynthetic pathways or for chemical evolutionary analysis and so on.